So some factors that are really important for supporting growth, we can, because they're so important for growth, we can flip this and use these to control, right? So things that are important that we need to maintain to grow an organism, we can take away or alter these things to control their level of growth, right? And of course, as it relates to bacteria, a lot of times we don't want them to grow, right? Especially when it relates to pathogens in our foods. So there's some, oops, I have no idea what I just did. Most organisms grow in fairly moderate environmental conditions, but there are some extremophiles out there, ones that just love and have adapted to really harsh environments. Do you guys remember a particular domain of organisms we've talked about that tend to be extremists? Archaea, right? But there are some bacteria that even fall into this category, although it's very rare for eukaryotes, right, to be extremists. So we need to know and be able to characterize organisms according to the environmental factors that are conducive to optimum growth. So what's important for growth? So this table summarizes that really well, right? And we're going to go through each section of this. So we're going to identify the influence of various environmental factors. What about water, right? How does having, not having, the problems you have with water activity affect growth? pH, which remember is the partial pressure of hydrogen ions, acids and bases, right? Do we have hydrogen ions? Do we have too much? Do we not have enough? Temperature, right? Is it too cold? Is it too hot? Oxygen concentration. Do we have enough? Do we have too much? Pressure, right? Atmospheric pressure. What effect does that have on cells? Radiation, right? So we're always asking, what influence does this have? But again, we can apply this information now regarding environmental factors that affect growth. How do we use this information to control the growth of organisms, right? So how does temperature affect, and then how do we use that information to control? So for water, as we know, we cannot live without. There is not a single living organism, including bacteria and archaea, that can exist without water. They cannot actively metabolize or grow without water. Availability and access to water can be limited by certain factors. Solutes being the most influential in limiting the ability of an organism to acquire water. We refer to this as the water activity, or sometimes the osmotic effect. The higher the solute concentration, so for instance, if you've got a salty environment, right, a salty water environment, the water is going to tend to want to stay with the salt and therefore not move into the cells. And so then you have what's referred to as a low level of water activity. And that's a really bad situation if you can't overcome it. If you can't get the water to come into the cell is going to die, right? If you're in what's referred to as a hypertonic solution, where you have a high amount of solutes. So obviously some organisms have adapted these types of environments. They've become what we refer to as osmotolerant. They can grow over a wide range of water activities. So as the activity goes from low to high to medium, they are okay. And how they've adapted is they have what's referred to as compatible solutes. This allows them to increase the internal osmotic concentration. They're compatible with metabolism and growth, right? But they're going to help with this problem of acquiring water from the environment. Because although the membrane divides the cell and makes these two separate environments, you have to get across that barrier. And if you're fighting against the concentration gradients, then you're going to have to expend energy in order to do that. And you don't want to have to do that unless you absolutely have to. And really, bacteria deal more with too much water, right? Water rushing in and potentially bursting their cells most of the time. That's why they have the cell wall that keeps them from bursting. So some of them have proteins within their membranes, right? Remember, that's the barrier, that require actually very high solute concentrations for stability and activity. So they've adapted to that other end of the spectrum. So when a bacteria cell is placed in a hypertonic solution, it undergoes plasmolysis. In this condition, the growth of the bacteria is inhibited, but not usually killed. Can anybody think about any type of hypertonic solution that we use to preserve our food? Salt. Salt is the most common solute we use to create hypertonic situations for bacterial cells. Meats are cured, right, using high concentrations of salt. This tends to draw the water out of the bacteria. Now, this is another term that I don't like because it's somewhat misleading. It says plasmolysis. It leads you to believe that what happens to the cell? It lyses. But in this case, no. It just shrinks. It becomes basically a raisin, right? Raisins are dehydrated grapes. You just suck the water out of them. Guess what happens if you stick them in water? They plump back up, right? And the same thing would happen to a bacterial cell. So although you may salt and cure your meats, if you rehydrate them, you'll rehydrate even the bacteria that may have been present there. The other problem is, are there pathogenic microbes out there that are resistant to salting? Yeah, staphylococci, we talked about growing mannitol salt agar plates, right? And then, Staphylococcus aureus, unfortunately, sometimes can produce toxins in our food that can cause food poisoning. And again, it's salt tolerant, so salting it isn't going to stop it. So is there any one perfect way for preserving in our foods? Not really. Sadly, no. Right? We do the best we can under the conditions we're in, but we also have to, watch for these situations where the organisms can overcome what we're using to try to control them. Right? So fungi are more resistant to osmotic shock than bacteria even. And then some bacteria are classified as halophiles. They've adapted to high salt concentrations. They actually require them, right? They've adapted so much to the other end that if it isn't extremely salty, they can't get water in their cells. So these guys are at 15 to 30% salt. Where on earth is this? Not the oceans. The oceans, believe it or not, are not that salty. We wouldn't be able to go in if they were. The Dead Sea, right? And the Great Salt Lake. Those types of environments.
It's so thick that you can float on top of it, right?
Yeah, yeah. So growth is going to be affected by that level of salt and how tolerant the organism is to those levels. So as you can see, right, non-tolerant, as you increase level, their growth rate is going to drop right off and you can even kill them. If it's one that's tolerant, right, you can increase the levels, but as you increase, its growth is going to decline, right? You'll get too high and you're going to kill them. And then you have these guys, as I said, that have adapted that, look, if you don't have enough salt, they can't even grow. It isn't until the concentrations get higher that they actually reach their optimum growth. And that's for your moderate halophiles, right? For your extreme halophiles, we're talking about that really salty environment. That 30% salt is when it's going to grow Before that, no growth is going to happen, right? And it's going to slowly increase as the salt concentration increases. Because they've adapted so much to that they actually can't get water in without that high salt concentration. Obviously, you know, they're going to thrive in those environments and not in the non-salty. They've adapted to that environment. Now, agar, which we talked about for our media, is a polysaccharide derived from marine algae, and we use it as a solidifying agent. It exists in a gel form, what's referred to as a colloidal solution. Therefore, it does affect osmotic pressure of the media. So typically, we don't go above 1% of agar in our media, but 1% is enough that you're going to get solidification, that you're going to get that solid matrix that we want to be able to grow the organisms on a solid surface. But if we go any higher than that, then we're going to basically start sucking the water out of our cells. And we definitely don't want it to do that if we're trying to promote growth. So the next is pH, which, remember, is the partial pressure of hydrogen ions. Hydrogen ions are released by acids into solution, and they're absorbed by bases in solution. Hydrogen ions play an important role in ionic balance, and as it relates to chemical processes. So some enzymes and stuff, if there are too many hydrogen ions, they interfere with their ability to do their chemical reaction. Others, if they don't have enough hydrogen ions, it interferes. So we have to maintain this balance. And the balance for our bodies in our blood is extremely important and is a very narrow range. 7.35 to 7.45 is the safe range of pH for our blood. If you go acidic, I can't remember which one it is. But I don't remember if that one's you go to coma or you convulse. Can't remember which way it is. Anyone named P2? Yeah, I haven't gotten to that yet. It's in the semester. All right. I don't want to tell you one way or the other, right? But if you go too acidic or too alkaline, right, one of two things happens. You either slip into a coma or die, or you convulse and die. Right? It's pretty serious. Yeah, I can't remember which one does what. If acid's the coma or alkaline is the coma. Basic. I don't know. Either way, it's not good news, right? That's the important thing, right? For A and P, you got to know the which is which. I can't remember. It's been too long since I've had to teach it. Someone's going to come up with a story, so I'll remember it, right? So, same thing for our microorganisms, right? They've got to maintain their internal pH. Now, how much the external affects them, right, is determined to what they've adapted to. But definitely ones that are pathogenic for us are classified typically as neurophiles, meaning they have to stay in that relatively neutral range, where that you have that balanced pH, not too much hydrogen ions, not too little. Okay, it's when we go acidic, right, that, as we know, is problems for us, but can be problems for microbes. So one of the things you'll notice here, as far as acids go, let me see. Who used vinegar to clean? Anyone use vinegar to clean? Vinegar is a very good cleaner. And again, the principle here is that you're affecting acid-base balance. In this case, you're using an acid. The problem is you don't want to mix some things, right? You don't want to mix an acid with a base, because what happens? You cancel them out. You create water and a salt. And then most of our soaps are alkaline or basic, right? So notice bleach, ammonia, soap, right? Again, why is this bad news for microorganisms? Because we're pushing them out of their comfortable pH range.
Someone please close the door, because I think it's going to continue for a little longer. And today I can't compete. Thank you. I appreciate that. So again, there's a whole list of organisms, right? Obviously, Staph aureus pathogenic for us, so, likes to be around neutral. There's a whole lot of bacteria, though, that can live in acidic environments, which probably makes sense why we have a lot of alkaline cleaners, though, right? Because they can resist acid, but not alkaline conditions, right? And we can push them out of their comfort zone and destroy them that way. So, unfortunately, these are a few numbers you do need to memorize. Organisms that are classified as acidophiles can usually maintain their optimum growth conditions between 0 pH and 5.5. So acidic, right? Low pH is acidic. Neutrophiles are going to stay around neutrality, right? So they can handle about 5.5 in the acidic range and up to about 8 in the alkaline range. Alkalinophiles can handle about 8 to 1.11 in the pH scale. 11.1. Well, 11.5. I'm alright. Not really, but yeah. Oh, I hate being sick. Okay. What's the highest pH? 14. Right? So these guys can't go the highest, right? And again, alkaline is probably the most limiting as far as environments. And again, it has to do with the access to hydrogen ions. So most acidophiles and alkalinophiles maintain, again, their internal pH is going to be neutral. It's the outside environment that they can resist those high hydrogen ion concentrations in acidic environments, and they can resist the loss of hydrogen in alkaline environments. And how they do this is they, of course, develop special proteins that have allowed them to resist the shock and the trouble that acidic environments can play. But because acid-base balance is really important, and as we've already seen, some of the bacteria, when they're exposed to sugars, are going to produce acid byproducts. They're going to put them into the environment. And if that organism keeps producing that acid, it's stuck in that petri dish, it can't get away from its own waste products, eventually it will kill itself. So to help with this problem and to get them to grow for this set amount of time that we're growing them in these closed culture systems, we typically add what are called buffers. And buffers help adjust the pH for small amounts of change. So if we're donating acid into the environment, it's going to help soak up those hydrogen ions and not have that environment be so acidic. If it's an alkaline type situation, it's going to donate the hydrogen ions to keep it from going too alkaline. But as I said, these can deal with relatively small changes. They can't deal with huge changes. So I always equate buffers to like the bumper on your car. It's going to help protect you and your car if someone bumps into you going about 20 miles an hour. Someone slams into you going 45 miles an hour, that bumper does you no good. It probably ends up underneath your car, like has happened to me, right? Small impacts, right? Little bumper, right? Little buffer is your bumper, right? So small impacts, not very large changes. So over time, when left in that system, they'll overwhelm the buffer system, and the organisms will die from their own waste. But to keep that from happening too quickly, we add buffers to the system to help adjust that pH. So what can happen to a cell if it can't maintain internal neutral pH? It's going to die. So how do we use pH to control microbial growth? What's a common pH changer that we use? Bleach, right? That's alkaline.
We even use it in food preservation. A lot of those organisms that produce the acids, right, then they die, but that acid that they produced in that environment when they fermented that food source we gave them impedes other microorganisms from growing as well, right? That acidic environment you've generated by fermenting those foods, right? Sauerkraut, right? Kimchi, all these fermented foods that are acidic and therefore sour, right? Because acids are sour, help preserve the food. Right? It's one of the main reasons why we would allow fermentation to happen, to create that acidic environment to help preserve our foods. Then sometimes we purposely add things, like purposely adding vinegar, which is also acidic. So for temperature ranges, the good news is you're not going to memorize the numbers. But we do need to know the classification from lowest to highest of the temperature ranges. There's 5 different optimum temperature ranges. And more important than temperature, we need to know some corresponding type environment where these organisms would thrive. And then be able to identify why growth rates increase with increasing temperature and then falls again at higher temperatures. So every organism has a cardinal growth curve where it has a minimum temperature in which it'll be able to grow. It has its optimum, its ultimate best temperature, and then it has its maximum. And as you can notice as it relates to growth, right, it really increases with increase in temperature. And that's because chemical reactions happen faster at warmer temperatures, period. But what happens when we get too hot? Yeah, what happens to those molecules? They denature, especially proteins. They get deformed by heat, permanently deformed, so they no longer function. 99% of the enzymes that catalyze the reactions that happen in our cells, including bacterial cells, are proteins. If you denature those proteins, you destroy those proteins, the cell can no longer function and will die. We use this knowledge all the time. What do we do to kill microorganisms in our food? We cook it. We cook it. You can eat it raw. If it's fresh and not contaminated, right? But our best bet and our best protection is to cook it. And then, of course, we've developed a like for that, right? Such that we don't usually want to eat stuff that hasn't been cooked. I still can't, I just can't eat raw fish. Just can't do it.
Once you let on us like a ceviche with the acidic outros.
Yeah, and so that's a good point, right? Ceviche, right, where they add the acids, right, from like lemon and lime juice, you're chemically cooking it. So you're still altering those proteins. Again, you're denaturing those proteins, but in this case, with a pH change. So again, it changes the texture, not exactly the same way that cooking does, that temperature does, but you're still creating that change. So, you know, I mean, whether you consider it raw or not, right, what does that really mean? But you are physically changing it and potentially destroying microbes in the process, because remember, if you go too acidic, you're gonna destroy them too. But that's why the texture of the meat changes, is because you're changing the pH. You're affecting those proteins. And it's a permanent change, right? Once you add that stuff, you can't make it go back the other way. Same thing when you cook something. So for our temperature ranges, mesophiles, their optimum range falls between 20 to 45 degrees Celsius. Their maximums are usually 15 to 20. I mean, minimums. And of course, this is Celsius, right? So most of us are like, oh, what? Right? So we'll do a little comparison and understanding of Celsius scale in a moment. But as you can see here, for some organisms where we know their minimum, optimum, and maximum, it makes sense that E. coli, Enterococcus faecalis, Neisseria gonorrhea, their range is 35 to 36 for Neisseria gonorrhea, and much warmer, of course, because I live in the internal, in the gut, of 37 for pathogens like E. coli. And the reason for that is that's our optimum core body temperature is 37 degrees Celsius. So it makes sense, obviously, for these organisms, that's their optimum as well. Because if it wasn't, they wouldn't survive inside of us, right? We maintain that environment. These guys, it's much cooler, right? But still within the range of 20 to 45 for optimum. Amoeba, right? Or no, that's not amoeba, it's a different one. It's a photosynthetic bacteria that's a mesophile. It's right here in the middle. Here's the amoeba, right? Amoeba proteus that can create problems for us. So notice, they like it a little bit cooler, right? That's their optimum. Their maximum is really outside of our range. But again, that's our core body temperature of 37. At your extremities, you're much cooler, right? And in certain parts of your body, you're going to have cooler temperature ranges. So again, with these guys, still within the range. Saccharomyces, right? This is why, making it warm will make them happy. So this is the one that makes your bread rise, right? Because it breaks down the sugars that you give it in the dough. And it'll produce acids too. But one of the other byproducts is carbon dioxide. And that's what makes the bread rise. And this is why when it's in a warmer temperature, it's going to happen faster because the organisms are going to grow faster. Simple chemistry. So let's look at Celsius scale for a moment. So Celsius scale is based on the properties of water. So at 0 degrees Celsius, what happens to water? It freezes, which for us is 32 degrees Fahrenheit, right? That's what we're used to.
That was negative 32 degrees Fahrenheit.
No, it's 32. 32. 32. Negative 32 is really freaking cold Fahrenheit. Yeah, you wouldn't want to experience that. Your refrigerator, you typically keep at about 4 degrees Celsius. Water boils at 100 degrees Celsius, which is 212 degrees Fahrenheit. At about 20 degrees Celsius would be what we consider room temperature of around 70 degrees Fahrenheit. And then 37 degrees Celsius is equivalent to human body temperature of 98 degrees Fahrenheit. And to give you an idea of how really hot 70 degrees Celsius would be, in Fahrenheit it would be equivalent to 158 degrees.
What's considered a low body temperature?
What's considered a low body temperature? I would not know off the top of my head. Okay, anything below 95. There you go. Ask our nurses. Okay, so psychophile. So phile means loving, right? Psycho, does anyone know what that means? Yeah, crazy. Which, as far as I'm concerned, if you like this climate, right? Because what is this, y'all? Water. This is just above freezing for water, right? So this is what? Cold. Right? So, as far as we're concerned, right, as far as I'm concerned, right, if you like cold, you're crazy, right? So this is cold. So, psychophiles, then psychotrophs, then mesophiles, meso in the middle, right, thermophiles, and then hyperthermophiles. So what type of environment are we talking about for these psychophiles? Where are we going to find them on Earth?
Antarctica.
Right.
at the poles, and not where it's frozen, but where you still have liquid water available, because remember, you got to have water available to live, right? There isn't stuff growing in frozen water, but there's stuff that will grow and then get frozen in water, right? So your glacial lakes and those pretty colors that you'll sometimes see when you travel to places like Alaska and to the poles, high up in Canada, and you see the glaciers and they have the different colors. There are microorganisms that grew when there was liquid water available. Algae and such will create those pretty colors in the glaciers. So when water is available, there is stuff that can grow at that really cold temperature. Do we have to worry about any of those making us sick? They wouldn't necessarily grow inside of us, right? Because we're too what? We're too hot. We're too hot. But if they produced any any toxins that we may ingest, then they could potentially make us sick. Although I don't know of any organisms classified as psychophiles that affect us or produce toxins that affect us. Psychotrophs, a whole different story, though. And these are the guys we have to worry about because they can grow at refrigerator temperatures, right? They can grow at room temperature. So these are the ones we really worry about potentially being in our refrigerated foods. The ones that are mesophiles, what happens to them when we refrigerate them? What happens to their growth rate? It decreases. It decreases. It doesn't necessarily stop. When we freeze them, we can stop growth. But does that kill them? No. Bacteria are very resistant to freezing. When you take them out of the freezer, they start growing again. This is why you don't want to freeze and refreeze your food, right? Over and over. I lost it. That's okay. It'll come to me. Mesophiles, right? So these are all the human pathogens that we worry about, right? Because this is the environment of the human body, falls under the mesophile classification, right there in the middle. Where are you going to find thermophiles? Yep, hot springs in Yellowstone National Park, right? Compost heaps. Are we going to have to worry about any of this stuff making us sick?
Maybe.
No. I think there's maybe one organism out there. And again, it would produce a toxin or something like that, and then we would ingest that or be exposed to it. Not that these organisms would be able to grow in us. We're too cold for them. Your hyperthermophiles, you're going to find these in the hydrothermal vents in the ocean floor. So literally living next to volcanic activity. Almost boiling water. So again, some of the hot springs in Yellowstone National Park are that hot. And yet you still have microorganisms growing there. So there's my picture from Yellowstone National Park. If you ever get a chance to go, go. It's amazing. It's like a drive-through safari and geological amazement. It is a crater left by a volcano. You will actually get tired of seeing buffalo. I stopped taking pictures of them. There's so many there. You get really excited when you see moose and stuff like that. Lots of caribou. Lots of stupid people. There was a whole gathering of people on the side of the road. And we're like, what's over there? They're like a bear. My husband and I got in the car and kept driving. Right? Everyone else was heading towards a bear. Not a good idea. Yeah, exactly. A lot of stupid people. But these pretty colors, y'all, microorganisms, right? And whenever you think you saw the most beautiful one, you go to the next section and it'll blow your mind. Yes. Yes, yes, yes. 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 Yes, because you guys are all packing up and not listening to me. So there isn't much left to this presentation, but what I am going to do to help you guys out
because I don't want to move the test back any more than it is right now, because the next unit is really big, is if I can't move the test, which I don't know. We'll see. I'm going to go find out about that today. I will post one of my recordings from a previous semester for the rest of this PowerPoint for you guys. Okay? For you guys to review over the weekend. And I'm out of time, am I? Okay. So this is one of the tough ones, I'll tell you guys, though. You definitely want to review and make sure you understand. Is the oxygen stuff, right? What can grow with oxygen, what can't, what levels of oxygen, and how are they protected, and why is it that these guys die? And it's because they have no protection from oxygen, right? And what are those enzymes doing? Okay? All right. The rest of it, believe it or not, you guys pretty much know already.